Welcome to AP Statistics. Today, we are exploring Topic 1.13, Experimental Design. Our learning objectives are to identify elements of a well-designed experiment and identify and justify types of experimental designs. By the end of this lesson, you will be able to distinguish between a simple observation and a rigorous experiment, and you will understand how to structure a study to ensure your results are valid and reliable. To understand how to design an experiment, we first need to define the 4 pillars of a well-designed experiment. These are the must-haves for any study that aims to show cause and effect. First is comparison. We must compare at least 2 treatment groups. Without comparison, we don't know if the outcome was caused by the treatment or would have happened anyway. Second is random assignment. This means using a chance process, like a random number generator, to assign experimental units to treatments. This helps balance out variables we might not even be thinking about. Third is replication. This doesn't mean repeating the whole experiment later. It means using enough experimental units in each group so that any individual differences among the units average out. Finally, we have control. We want to keep potential extraneous sources of variation the same for all groups. If we are testing plant food, we should make sure all plants get the same amount of water and sunlight. A critical tool in the comparison pillar is the use of a control group. The control group provides a baseline to see if the treatment of interest actually has an effect. Sometimes, this group receives a placebo. A placebo is an inactive substance or fake treatment that looks like the real thing. The placebo effect is the difference between the average response to a placebo and the average response to no treatment. The placebo effect occurs when subjects show improvement simply because they believe they are receiving a treatment. By giving the control group a placebo, we can account for this psychological effect. To prevent bias, we often use blinding. In a single-blind experiment, either the subjects or the researchers don't know who is getting what. A double-blind experiment is the gold standard. If neither the patient nor the doctor measuring the results knows who got the real medicine, it prevents expectations from influencing the recorded data. Now let's talk about why we randomize. The goal is to avoid confounding variables. Imagine a study on whether diet soda causes weight gain. If the people who drink diet soda also happen to exercise less, we won't know if the weight gain is from the soda or the lack of exercise. In this case, exercise is a confounding variable. An extraneous variable is a variable that affects the response but is not the main one being studied. We use direct control to handle variables we know about. For example, in a lab, we keep the temperature constant. But for the variables we can't control or don't know about, we use random assignment. When we assign units at random, those hidden variables like metabolism or genetics should be spread roughly equally across all groups. This balances the groups so that the only significant difference between them is the treatment itself. Now that we understand the ingredients, let's look at the 3 main types of experimental designs you need to know for the AP exam. The first and simplest is the completely randomized design. Treatments are assigned to experimental units completely at random. In this setup, every unit has the same chance of being in any group. We might use a hat or a random number generator to split 20 students into 2 groups of 10. While the groups don't have to be exactly the same size, it is often preferred for statistical power. This design is best when the experimental units are relatively similar to start with. The second design is the randomized block design. First, group units by a similar trait called blocks. Second, randomly assign treatments within each block. Think of a block as a way to group similar units to reduce variation. For example, if we think a new medication might affect men and women differently, we would block by gender. Total subjects are split into a male block and a female block. In the male block, we randomly assign participants to treatment versus control. Likewise, in the female block, we randomly assign to treatment versus control. The purpose of blocking is to separate variation caused by the blocking variable from the response. By comparing men to men and women to women, we can see the effect of the treatment more clearly without gender clouding the results. Remember, you block what you can't control but can identify. The third specific type of design is the matched pairs design. You can think of this as a special case of a randomized block design where the block size is exactly 2. There are 2 ways to do this. First, we can pair up 2 subjects who are very similar. We match them on a variable that might affect the response, like age or health status. Then, for each pair, we flip a coin to decide who gets which treatment. The second method is to have the same person receive both treatments. For example, if we are testing two different types of running shoes, a runner might wear shoe A for 1 week and shoe B for the next. Crucially, the order must be randomized so that fatigue or learning does not become a confounding variable. The goal of matched pairs is to make the comparison as precise as possible by controlling for individual differences. It is often much more effective than a completely randomized design because it eliminates the noise of person-to-person variation. Let's wrap up by reviewing our learning goals and seeing how they apply to a real-world scenario. We have covered the 4 pillars of design: comparison, randomization, replication, and control. We also looked at blocking versus matched pairs and the importance of blinding and placebos. Imagine a researcher wants to test a new fertilizer on tomato plants. In this scenario, we have 60 tomato plants. However, there is a potential confounder: sunlight exposure. If the researcher just puts the fertilizer on the 10 plants closest to the window, the results are confounded. Was it the fertilizer or the extra sun? To fix this, a well-designed experiment would, first, block by sunlight; second, randomly assign fertilizer or a water control group within each block; third, use all 60 plants for replication; and finally, control variables like water and soil type for everyone. By blocking, the researcher accounts for the sunlight variable. By randomly assigning, they balance other variables like plant genetics. By using a control group, they have a baseline. And with 60 plants, they have enough replication to ensure the results aren't just a fluke. Let's check our goals. Did we identify elements of design? Yes. Did we identify experimental designs? Yes. And did we justify the design choice? Yes. You now have the tools to identify and justify a well-designed experiment. Whether it's a completely randomized design, blocking, or matched pairs, the goal is always the same: reduce variation and isolate the effect of the treatment.